Welcome to Cancer HealthCast, where science is driving hope. I'm your host, Nikki Henderson. Today, we are joined by Dr. Eytan Ruppin, who is the founder and chief of the Cancer Data Science Department at the National Cancer Institute. He will be talking about a new artificial intelligence tool that's helping to better match cancer patients with specific therapies, as well as give details about the NCI CCR Synthesis Clinical Trial that will begin later this year. Hello, Eytan. Thank you so much for joining us on the show.
Hi, Nikki. It's my pleasure.
Well, let's go ahead and jump into our conversation. Eytan, can you start off by telling me more about your current role and the overall mission of the Cancer Science Department?
Yes, with pleasure. So I was hired and brought into the NCI in 2018 by Tom Mistelli, then the director of CCR, NCI's Center for Cancer Research, to set up and found a new branch, what we call a department, that will be focused on analyzing cancer-related data science. And as you may know, there is in the last decade and even more, there is very fast advances, very large scope advancements in collecting molecular data from the tumors, what we call the omics data, different facets of molecular and genomic data, in order to, first of all, better characterize the tumor's landscape, their evolution, understand better on basic science aspects, what's going on in tumors, what makes tumors tumors, what determines their aggressiveness, what molecular alterations are responsible, associated with the likelihood to metastasize and become more aggressive. So that's from the basic science side. And from the clinical side, the goal is to collect all this data in order to improve our understanding, first of all, our ability to predict new cancer vulnerabilities and cancer treatments, and second, to identify them, and second of all, to develop biomarkers, which will enable us to better predict the response of patients to different possible treatments and to match each patient to the best treatment that could help him on the long run. So a lot of data is being generated in the last decade in a quite unprecedented scale, mainly due to the advent of sequencing technologies as mass spec, and other omics technologies. And that creates an immediate and present need to develop approaches to analyze, computational approaches to analyze the data and try to make a sense of it, try to see the interesting trees from the forest of data. And that is done, of course, nationwide and worldwide, but the NCI felt a need to have such a department in-house in our center because, our center, of course, it's more than 200 cancer labs and so on, is creating a lot of such data, both for basic research and in our clinical trials, in our clinical center, and so on and so on. So that's why I was brought to found this department.
Oh, wow. Very interesting. Well, what are some of the most recent genomic approaches the clinic is utilizing to better match patients to specific therapies?
Yes, sure. So currently, most of approaches are genomic approaches, and based on analyzing the DNA, what we call DNA sequencing, to find genomic events. It's mainly mutation in major genes that are responsible for cancer, that are called cancer driver genes. And some of these mutations, when they occur, they are actionable. What we mean by actionable is that there are drugs that have been developed over the last 2 decades, and mainly in the last decade, that are designed to specifically target these mutated cancer driver genes, achieving more selectivity in our ability to hit cancer and hence more effectivity at times than standard chemotherapy and so on and so on. So there are various ways of sequencing the tumors from biopsies or from post-surgery specimens to identify such actionable mutations, and this guides us for treatments, for a specific class of treatments, which are called targeted therapies. In addition to identifying these actionable mutations, DNA sequencing, or what we call the sequencing panels, also enables us to estimate what we call the TMB, the Tumor Mutational Burden. That is one of the strong biomarkers of the likelihood of response to immunotherapy, which, as you may know, is another very promising avenue that has been developed in the last decade. So genomic sequencing tells us a lot, but it doesn't tell us the full story, regrettably, because only for about 10 or 15%, 15% of cancer patients can we identify such actionable biomarkers. So there is a lot of space of trying to develop to look at other types of molecular tumor in the data and see if we can develop biomarkers that can help us guide the treatment for the large fraction of patients which are not getting currently recommendations just by looking at these DNA sequencing panels. Is that clear?
Oh, certainly, yes. Thank you so much. You explained that perfectly. Thank you. Can you talk about the efforts that are underway to develop bulk tumor transcriptomics? And can you also explain a little bit about how this method relates to the NCI CCR Synthesis Clinical Trial that is expected to start in the fall? And what role will it play in treating aggressive breast cancer?
Yes. Let's try to break it down to a few sub-questions. So, as I said, there is a clear and present need to look at other types of omic data to help more patients. So one very promising type of molecular look at is the expression of the genes. You see, when you look at the sequencing mutations, that's very important, but that tells you what happened in the exact, code of the genes and so on. But actually, that's just the DNA that is passed. But some of these genes may be expressed or highly expressed in some tumors, and some of these genes may not be expressed. That means that even they may have DNA alterations, they are not consequential because they may not be expressed. They are not part of the cohort of proteins that are actually expressed in the tumor cells and orchestrating their behavior. So to gain more insight on the latter, one way to do it is RNA sequencing, which quantifies which genes are expressed and to what level in each tumor. And then others and many others in the world, and also our group, has been doing efforts to examine how we can look at this type of data and stratify patients to the best treatments. And of course, I'm not going into technical details, but there is a lot of publications and peer-reviewed publications on that from others and from my group. For interested listeners. But lo and behold, we think that we can help a sizable fraction of patients to match their treatment, to best match drugs for the treatment beyond the recommendations that are made by the current standard of care, at least in big US medical centers of DNA sequencing. We are hoping, we don't know, it needs to be studied prospectively, and I want to be careful and cautious. We are hoping that we can increase the number of patients that can benefit from useful recommendations from the current 15% to, let's say, 30 or 40%. But that remains to be studied very carefully. So this is to answer the first part of your question. Indeed, in this line, to answer your second part of the question about the synthesis trial, so this will be, to the best of my knowledge, the first prospective trial that harnesses tumor expression data to try and treat aggressive breast cancer. So there is a form of aggressive breast cancer that is called TNBC, triple negative breast cancer, that currently doesn't have very good treatment options. And in this, or no ways to biomarker to assign these treatments effectively. And in this trial, which be led by our women's malignancy branch in the NCI by Stan Lipkowitz and Sheila Rajagopal, we will look, aim to treat such patients and choose the best treatment that we can find to these patients out of an array of 16 different possible targeted and immunotherapies in the scope of breast cancer. And this is an excellent example of the kind of trial that, can almost only be done in the NCI, because it involves many different treatments, and it's a quite complicated trial to run. And it's a trial that will not be supported by big pharma and so on, which mainly support, one drug trials that may, promote and advance and show the value of the specific drugs that they are developing. So it will take some time. A prospective clinical trial takes some time, but we are crossing fingers that it may be successful and helpful.
Well, I certainly hope so. I'm definitely going to cross my fingers as well, because it does seem very, very promising. Well, Eytan, what is the difference between bulk and single cell when it comes to determining tumor heterogeneity?
Right. So let's start from the beginning. One of the challenges of modern therapies is that they are very helpful. And for sometimes, we get the ideal result we are hoping for, but we are keeping the disease in check for many years. We actually, in some cases, think that we even have cured the disease and so on. But in other cases, regrettably, even though initially we get very good responses, and that's very important, after some time, resistance may develop, and the tumors may overcome the treatment. So then we need to think of other treatments and other options and so on. So a lot of people think that a major force in underlying this emergence of the resistance is that we are only treating some clones, some parts of the tumor cells, and we are not hitting all of them. So by killing the clones that are responsive to the specific treatment, we actually give way and space to the rise of other clones, which eventually cause resistance. So people think that, one promising way forward is to have combination therapies of specific treatments that targets as much as possible many clones in order to reduce the likelihood of resistance. But to do that, you need to understand what are the different clones, tumor clones that are composing the tumor and its microenvironment. So bulk sequencing, either it's DNA or RNA, you can think of it as just one snapshot from the tumor, from where you are taking the specimen and sequencing it. So actually, you cannot understand the overall clonal structure. You can do it by more modern advanced sequencing techniques, which are called single cell sequencing, DNA methylation RNA, where you can actually sequence in one stroke and understand the expression of tens of thousands of individual cells, both tumor cells and immune cells in the tumor microenvironment. Doing that and clustering the cells and so on, you can enable you to understand the tumor clonal structure, its heterogeneous structure, and think of matching combinations of treatments that we are hoping will be more effective and help prevent or delay the emergence of resistance to therapy.
I see. Well, thank you so much for breaking down the difference. It's much more clear now. And like you said, I hope that this will increase the likelihood that you can determine or reduce the resistance of the cells so that these tumors won't keep multiplying and regrowing. Well, Eytan, we're going to shift the conversation a little bit to talk about artificial intelligence, which is making headlines daily. There was a recent study that was published about a new AI tool developed by NIH scientists. And I wanted to talk with you and ask you, what is this tool and how is it helping cancer patients when it comes to their treatment as well as other benefits?
I assume you are referring to a study published by my lab recently in Nature Cancer. Is that the one?
That is the one. That is the one.
Yeah, because there's many fantastic studies by others, and ours is just one of them. So the idea is, again, be able, trying to harness AI and machine learning to analyze big data from patients and tumors and from the patients themselves in order to assess the likelihood of response to treatments and hence tailor the best treatments. Now, this is a very serious challenge, and many groups are working on it, including ours, and there's a lot to be done. Actually, until now, we were talking a lot about sequencing and molecular profiling, which is indeed very important, but still has its costs and so on and so on. So in the specific study that you are referring to, which was done in collaboration with Luke Morris's lab at Memorial Sloan Kettering, we actually took a different angle. We said to ourselves, to what extent can we predict the response of cancer patients across many indications to immuno checkpoint therapy, which is now a major type of treatment. Mm-hmm. And we analyzed very large thousands of patients' data from MSK, and we actually did not look at complicated molecular transcriptomics data. We actually went back a bit and we just looked on simple clinical characteristics of the patients, on variables that are routinely observed in blood tests in the clinic. And one additional factor, this TMB, this tumor mutational burden, which gives us just a quantitative estimate of the total load of mutations in the cancer driver genes. So no complex omics data, really back to the foundations. And we find that overall, we get quite a reasonable and clinically helpful, I think, predictor of the patient's response to immune checkpoint therapy across many indications. So to give a number, it will not say much, but about 75–80% accuracy. And just from these simple, clinically readily available variables. So we also made it available to the public. So there is a calculator that is posted on an NCI designated and approved website as a service to caretakers and possibly to patients. And everyone can access it and enter a few characteristics that characterize the case that he's interested in, and get an estimation as best as we can of the likelihood that he will respond to immunotherapy. So we are hoping that this may be helpful for some.
Yes, I really hope that it is. And Eytan, can you tell me some of the other areas of oncology where AI is being applied, and in what ways is AI helping to advance clinical data, imaging, and precision medicine overall?
Yes. So AI is currently applied in two main domains in cancer research, and each one of them actually relies and applies a different type of AI/machine learning methodology. So the first one is all this world of electronic health records, all the vast information that is collected upon us and so on from our doctor visits, hospital visits, what have you, and so on and so on. And, huge troves of data, and many academic labs and companies and so on are using what is called LLMs, the large language models, in order to mine this data and try to develop useful applications. So all of you know what's ChatGPT and the like. So these are large language models. So you can train these models specifically on medical data, medical textbooks, electronic health records, and so on and so on. And then you get specific, if you want, cancer-related AI agents that you can probe and consult with, and so on and so on. So there's a lot of interest and activity in that space. For example, a group of researchers in CCR, in NCI, has recently, which I was a small part of, has recently published a paper in the New England Journal of Medicine AI, testing the ability of such LLMs to answer questions in oncology, and compare it to the level of oncology residents and so on. So, ideally, we would like to have such tools that could help at different levels patients and clinicians and oncologists to ask questions and consult with based on the whole knowledge. And it's very interesting to see where it will go and to what level it will succeed. The other domain, which has been seeing striking advances, is harnessing AI and machine learning to analyze images in medicine in general, and specifically in cancer research. And one of them is radiological images, CTs, X-rays, MRIs, what have you, and help the doctors identify and the interesting, and locate and identify and label the interesting findings in the images. And the second domain is that of the 100 years good old, what we call H&E, hematoxylin pathology slides. And again, the machines are showing really exciting results, not to replace radiologists or pathologists, but to actually help them do their work much more effectively and accurately. And my lab in the last 2-3 years has been very interested and active in harnessing these deep learning approaches to analyze pathology slides. So what we have been doing in the pathology space, and this is in close collaboration with my good friend and close collaborator, Eytan Ruppin, who is Chief of Pathology at the NCI, has so far been focused on two things. So we have discussed that we have developed approaches to tailor treatment to cancer patients from the measured tumor transcriptomics. So what we did in the last few years, and it's another paper that is soon to be published in Nature Cancer, we asked ourselves and answered positively that instead of measuring the tumor transcriptomics that takes a lot of time and costs considerable money, we asked ourselves, can we predict and infer the tumor transcriptomics from the slides? And if we can do so, then we can, we have already approaches to, quite reliably recommend treatments to therapy. So that's what we did. That's what we succeeded in, and I'm very excited about. So in other words, we can now have a pipeline that can take the pathology slides and analyze it and give you treatment recommendations in a very speedy and low-cost effective manner. Of course, we are showing it retrospectively on independent data sets. We show the value that will be in the upcoming paper. But anything that is shown, in clinical and in cancer research should be carefully and rigorously tested in prospective studies. And that has still not been done, but hopefully soon. The other thing is that other groups in the world, and we have also joined that, we show, but just by looking at the slides, we can actually help the pathologist classify tumors at a speedy, high-resolution manner. We did it in brain tumors, which is a very challenging task, and our predictors have accuracies of around 95%. And actually, at the level of pathologists, neuropathologists, expert neuropathologists, or perhaps even better.
Okay, perfect. Well, are there any future goals or initiatives that you're excited to talk about?
Yes. There's many, we are doing our poor best, but there is so much that needs to be done. But particularly in lieu of time, I really want to focus on the efforts to develop ways, very cheap and fast ways, to recommend treatments to patients from the slides. That's the thing that currently excites me most, and we are investing a lot of effort in, and I'll explain a bit more. First of all, in the US, if we can do it, if we can deploy such systems, we can bypass the bottleneck with current molecular omics-based treatments, and that is that they cost considerable amount of money, not too much, by the way. We can talk numbers, but considerable amount of money. But the other thing is that they take time. Even in large medical centers, they take to get the results anywhere between 4 to 8 weeks. Sometimes the oncologist and the team that treats patients doesn't have that time. They want to start treatment early, and they would like to have recommendations early. Slide-based treatment recommendation systems, if we can further develop and validate them, could do it in 1 or 2 days. So it's like almost immediate. It's a game changer if we can work it.
Yeah.
The second thing that I'm gung-ho about is that potentially such systems could be deployed in the developing world, in under-resourced regions who do not do sequencing and will not do sequencing, in the foreseen future. And that could be a game changer for precision oncology in the developing world. So it could potentially help a lot to better equate and democratize cancer treatment, either in underserved regions in the US, if we start from our own home, but beyond that, in the developing world. So that's, a direction I'm very excited about. But beyond that, there are additional exciting domains. So perhaps the most exciting thing my lab is currently working on is on predicting spatial transcriptomics. So I don't want to bore you with the details, but this is now measuring spatial transcriptomics enables cancer researchers and clinicians to understand the not only the tumor overall, but the intricate architecture of the tumor microenvironment, which is very important, again, to clinically how we can treat treatments. The experimental ways to measure and assess spatial transcriptomics and spatial biology are still very costly. And we believe we actually have very encouraging preliminary results that we can infer that, again, not only the bulk transcriptomics, but we can infer the actual spatial transcriptomics of the tumor from the slide. And if we could pull it off, as I hope, I believe to be quite transformative. In closing, looking at the future, I want to say a few more words on a higher level. The key for success of data science in advancing cancer research and treatment is the analysis of big data sets. Okay? Mm-hmm. And that critically depends on the making such data available for academic use, at least for everyone across the world. And there are steps that are being taken, some of them, orchestrated and mediated by the NCI, but not enough, in my mind, is being done. Much more should be done. There are challenges concerning very important issues of patients' privacy and security. Of course, they need to be taken care of, but I think that more could and should be done to make much more data available to academic researchers in the US and across the world, because without that, we will not be able to make the advancements that we wish to make. There is also an issue now in the whole AI research about the balance between academia and the large companies. So, as you may know, large company players in the AI field have much more resources, money, human resources, access to data, potentially that money buys, than academic centers. That is great, and of course, we welcome anyone, but we should think about it carefully, because we want that a considerable part of the research will still be done in academic institutes and thus made publicly available to everyone, not siloed, behind some companies' borders and so on. And that's another topic that requires careful attention, and I think about a lot. So there is a bright future, really, and would be very, very interesting coming few years, because AI is really transformative. But we need to carefully come with a plan to make the data more available for public use by research centers across the US and the world.
Yes. Thank you so much for sharing all these future initiatives you're excited about. I'm excited about it, and I hope that down the road, it would be great to touch base with you again and get some updates, especially how AI is impacting precision medicine. That's wonderful. Well, I want to tell you that I really enjoyed our conversation, and I appreciate you taking out time to speak with me today about AI and how it is impacting precision oncology. So thank you again, and I really hope that you have a wonderful afternoon.
You too, and thank you so much for having me.
HealthCast, along with GovCast and Cybercast, is a production of GovCIO Media and Research. To explore our content, visit our website, govciomedia.com. Keep an eye out for new episodes every Tuesday. And if you like what you heard, leave us a review on the podcast platform of your choice. Have a topic you want us to discuss? Contact us at newsletter@govcio.com.